reactants. So this one would be the best. That's the line that shows that distance, so that would be D. Number 2 says, which of the following represents the activation energy required for the enzyme catalyzed reaction in the figure? So you can see there's 2 peaks here. Which distance is showing the activation energy for the enzyme catalyzed one? B. Everybody agree? Yeah, so it is B. It's going from the energy of the reactants up to the energy of the peak, but for the enzyme catalyzed reactions, still it has lower deactivation energy, so it should be letter B. Okay. Which of the following reactions would have an energy diagram similar to the one shown above? And for this, we just circle all that apply. So this is like a sticky notes that we did yesterday. And basically, we can start just by saying what type of reaction is shown here. Is it energonic or exergonic? Exergonic. So we're just identifying which of these are exergonic reactions. So conversion of starch into glucose, is that exergonic? Yeah, so that one would be exergonic. How about phosphorylation of ABP to create ATP? No. Hydrolysis of a polypeptide chain. We're hydrolyzing, right? So we're releasing energy. Synthesis of a nucleotide. No. Glycogenolysis or conversion of glycogen to glucose. Yes. And then generation of a triglyceride molecule. No. So I didn't realize they were all down the line. So A, C, and E. All right, so for number 4, there could be a couple of different terms. I think yesterday I used several different terms. But when you have an end product that feeds back and inhibits an earlier step in the pathway, what is that called?
I put negative feedback loop because I couldn't remember what.
Yeah, so that's fine. I would accept that because I think I said that yesterday. I might have said like, yeah. Any others that you thought of? Yeah, feedback inhibition I think is what the textbook says, but I would accept that because yesterday I said a whole bunch of different things. So that's kind of what I was thinking of, but again, if you said negative regulation or negative feedback or something like that, I would accept that. Okay, and then number 5 says, give an example. Can be from anything we talked about yesterday, of an enzyme or a step that's regulated through the mechanism you mentioned in number 4. So we talked about a couple examples of this, but does anybody want to share the one that they wrote down? No? You're like, no, I don't want to share.
I couldn't remember exactly the name of everything, so I wrote down the general idea. It's like when there's enough ATP floating around, then the ATP binds to something else in this enzyme, and it kind of creates a conformational change so that whatever is supposed to bind to the enzyme can't.
Yeah. So you could give that example. That one was from glycolysis, so if you described it like that, I'll give you credit for that. That one was it was PFK, but again, I told you didn't need to know the names of the enzyme, so even if you just said early enzyme in glycolysis is inhibited by ATP binding at a different site, something like that. That was one example. There's another one we talked about with pyruvate dehydrogenase. Again, you don't have to know the name of the enzyme, but that was in pyruvate oxidation. And remember that high levels of ATP make it so that that gets phosphorylated and then it's inhibited. So if you thought of either one of those, we also talked about in TCA cycles, some of the early enzymes are also inhibited by having too much ATP. So if you mentioned any of those steps, then that would be right. Any questions about number 5? Okay. Let me show this so you can notice it. Okay, so then number 5 says, in some of the reactions you performed in lab, well, specifically when we did the plasma purification, we used EDTA to bind divalent ions. So remember that was a chelator of divalent ions like magnesium, and the purpose of that was to deactivate enzymes. So what do the divalent ions do? So which answer was the best answer here? Things that inhibit the enzyme, or they don't inhibit, they help the enzyme. Yeah, cofactors. The difference, what is the difference between cofactor and coenzyme? They're both things that bind to enzymes and positively regulate them, but can anybody remember the difference between a coenzyme and a cofactor?
Well, the one's ions, I don't know what the other one is.
Yeah. So one is inorganic, so cofactors are inorganic, so they could be like ions, and then coenzymes are carbon-containing or organic compounds. So like our vitamins, like vitamin B, all of those act as coenzymes to energy enzymes. All right. So the number 7, this is kind of getting at, we talked a lot about oxidation reduction and being able to identify which one is oxidized, which one is reduced. So we're circling the reduced compounds. So we have on one side malate, the other side has oxaloacetate. Between these two, which one is reduced?
Malate.
Yeah, so you notice there's more hydrogens here. This has double bonded oxygen. That's really the difference. So this one is reduced. So circle this one. On this side, there's something else that is reduced. It's not oxaloacetate, but what else is on this side would be reduced? NADH. This has the electrons. And then on this side, now going to the oxidized, this is oxidized, and this is oxidized. So essentially the electrons are being passed from malate to NAD+, and then on the other side of the reaction, NADH is now reduced, and oxaloacetate is now oxidized. Yeah.
Would H plus be reduced technically, or just hanging out?
Yeah, that's a great question, if that is considered, because that probably came from here, right? Great question. I actually would ask a chemist, is this considered? If you put it or not, I'll just look at these, the main compounds there. What did you put, oxidized?
I said reduced.
You said that it's reduced because
H
it's,
plus.
oh, but I guess H plus is just the proton, so it's
Yeah.
missing its electrons.
Yeah.
So if anything, maybe it should be oxidized.
Yeah.
Okay. We'll just worry about the main transfer of electrons there. All right, so number 8 is the chart for outputs of glycolysis. So for the net ATP, what number did you put? 2. And then NADH, 2, CO2,
Yeah.
and then FADH2.
Yeah.
Okay, so 2, 2, and 0, 0. And then the last question is just kind of just regurgitation of what we learned about ATP production. So what are the 2 types that we talked about yesterday? Substrate level and an oxidative phosphorylation. So, and then just explaining the difference. So hopefully in there you kind of define that substrate level uses a high energy phosphate and is enzyme catalyzed and transfers the high energy phosphate to ADP to create ATP. Oxidative phosphorylation, main thing is that it uses ATP synthase and an electrochemical gradient to produce ATP. So something along those lines. Okay, so that's our last quiz. So anybody have any last questions about kind of the energy enzymes or so far what we've done with the cell respiration? All right, so today, since we're going to be talking about electron transport chain, if everybody can take it back out, this poison case study. I don't know, did you get one covered? You have these. Oh, you have extra copies, okay. You can take those. Okay, so don't read the back of it. This is because it's a surprise what maybe some of you remember this. Or maybe I'm the only one who is old enough to be alive. I was only 1 year old, but at the time of this poison, so this is based on a true story. So this happened in Chicago. So if you kind of read the top, there was a couple of cases where people died unexpectedly. And so that Mary Kellerman gave her 12-year-old daughter a painkiller, and she woke up complaining of a sore throat. And the daughter died later on. Adam Janis, postal worker, also died unexpectedly, and they thought he had suffered a heart attack. And his brother and sister later became ill and died. And then 3 more unexplained deaths occurred in the nearby Chicago suburbs. And what was kind of linking all of them is that they had all taken Tylenol before their death. So does anybody remember this or has heard of this before?
I was told about it as well too.
Okay. So the symptoms that they had in common were weakness, dizziness, fatigue, sleepiness. They were flushed, bright red skin, headache, shortness of breath, vomiting, confusion, and disorientation. So for question number 1, I just want you to kind of talk with people nearby and jot down some ideas here. So are they 7 deaths connected? If you were doing this investigation, what information would you need to figure out to kind of connect them all together? So you can go ahead and work in pairs or groups of 3 and just do like bullet points, what you think you would want, what kind of information would you want to collect. And then you can go on to part 2, which is looking at the autopsy report. And you might have to look up some of these terms on your phone. So feel free to, or your laptops, look up hypoxia, look up what the normal oxygen level in the